Hello to the biochemists out there. Welcome to lecture 9 of BIS103. And today we're actually leaving our discussion of carbohydrates for a little bit. And we want to focus on another major metabolite class that we'll be covering this quarter, which is our fats and lipids. And we want to look into the metabolism of these compounds. And two major topics that we'll be covering is glycerides, how they're being broken down, and the specific mechanism of Becker oxidation and how we can catabolize fatty acids. I've broken up this lecture into 3 videos. This first one here will focus on the chemical properties of lipids and fatty acids. And there will be another one on catabolism and the mechanisms of catabolism. And a third short video, another one of our vitamin Bs, our cofactors, that I will cover there, vitamin B12. So watch out for those. For this video, our learning goal will be we want to understand the biochemical properties of fatty acids and lipids and what makes these compounds such an important source of energy as well as the formation of membranes. There are several reasons why lipids and fatty acids are so important. One of the major ones highlighted right here is membrane integrity. So fatty acids, because of their chemical properties that we'll see in a little bit, are really critical for the formation of membranes. And I'm sure you're all aware that without membranes, there would be no life. So this is obviously a very essential functions of our fatty acids here. The second one right here, which people are often less familiar with, is that especially free fatty acids can also function as signaling molecules. One example I highlight here are prostaglandins. These are actually lipid derivatives that function as signaling molecules, for example, in processes of inflammation, pain response, and so forth. And then the third role here, and that really is, you could argue, the major one, is the central energy resource. So the bulk of our fats and lipids we're actually using to generate chemical energy, and we'll see how this actually works in comparison to our carbohydrates. So focus today will be on this role here as a central energy resource. And for that, we need to first understand the chemical properties of fats and why they are such a good resource. Okay. To do so, we just have to go back all the way to lecture 2 and remind ourselves of some of the common functional groups that we had discussed and how they relate to the reduced and oxidated states and how this in turn relates to energy. Right? We had talked about these functional groups and we had seen many of those, all of those in fact, in our discussions on carbohydrate metabolism. Right? We had our alkanes here, the simple hydrocarbons, as the most reduced functional groups. And then we had continued to oxidize those through an alcohol function, an aldehyde function, a carbonic acid function, and eventually carbon dioxide as our most oxidized form. And if you remember lecture 2, we had also spoken about the energy in these functional groups. And we had said, right, the most reduced groups here have the highest energy. The energy is trapped largely in the electrons that are contained in these bonds. And the more we oxidize those, the more we lose these electrons, we are going to a lower state of energy. And now this becomes really important when we think about the energy that is maintained in our fats. Okay. So for that, we have to look at sort of a general structure. What actually are fatty acids? Fatty acids essentially are long chain hydrocarbons here. So a lot of these alkane functional groups with a terminal carboxy group. Right? So a pretty simple molecule in itself, a lot of carbons. We can have everything from 2 to 34 of these methylene groups here. And what we want to do first now is to compare them back to our sugars and understand what kind of functional groups do we see here and how does this relate to the energy in these metabolites. So let's compare fatty acids and carbohydrates and go through these functional groups one by one to see what we have. Right? If you look at alkanes, yes, we have them, right? I showed them just right up here. Here, all your alkane groups. If you think about the carbon backbone of your sugars, I'm not showing the sugars here. You should be pretty familiar with the structures by now. We also have alkane functions there. Then alcohols. Sugars, yes, right? We had talked about the definition of sugars. We have polyhydroxy, that's part of our definition of a carbohydrate. Obviously, we have hydroxy functions here, but if you actually look back at our fatty acid structure up here, we don't have any alcohol functions there. Right? What about aldehydes, right? Our other definition of the carbohydrates was, right, there are either ketone or aldehyde functions in there. So yes, we have aldehyde or ketones in our sugars. Again, if you look up here in your fatty acid, no aldehyde or ketone to be seen. So this functional group also is missing in fatty acids. The opposite now is true for the carboxylic acid, right? We have our terminal carboxylic acid here in the fatty acid. But right, if you remember our sugars, none of these have acid functions. We saw some acids, right, in, for example, our PPP, but they don't meet our definition of a carbohydrate, of an aldehyde or ketone function. It's then a derivative of a sugar, but not a sugar. So sugars do not have carboxylic acid functions. So what we see here, right, is that our sugars have a much larger variety of these functional groups than fatty acids. So you could argue, why are then sugars not really the better resource and the major resource? Why is it the fatty acids? And the reason really is, if you look back at our structure up here, right, we have these alkane functions and we have many of these. And if you now look at that, these alkane functions are the highest in energy, it starts to make sense why fatty acids might be such a good energy resource, right? They have a much larger number of these energy-rich alkane functions. And so what we would say in this case is that fatty acids have a much higher energy density as compared to sugars. In fact, it's about twice as high. So fats are actually your major energy resource. Okay. It sounds counterintuitive. We've all talked about the sugars and how important they are for generating chemical energy, and that is true. Right? Fats are higher in energy, but we will actually see in this lecture that breaking down fats, lipids, and fatty acids is actually a bit slower. It is metabolically more demanding and more complex. So using both sugars and fats for the generation of chemical energy makes a lot of sense because they're complementary to each other. Fats are a little higher in energy density than sugars, but sugars are actually more readily available. So you can think of it in using energy over time, right? Sugars are basically a fast response, getting energy quickly, but fatty acids and lipids can be stored much better and are more energy rich, and so a really good energy resource over the long run. Okay. Both are really important though. So now to understand fatty acids, we have to start looking into their structure and their chemical properties. So let's start with a little bit of nomenclature. We are comparing saturated to unsaturated fatty acids. Saturated fatty acids are those fatty acids that do not have any double bonds. Okay. So I highlight the structure here of a saturated fatty acid, right? You have your long chain hydrocarbon and your terminal carboxy group. If it's easier for you, we can also draw it like this, right? Here's your hydrocarbon chain right here with your terminal carboxy group. You typically are starting to count the carbons from your terminal carboxy group. So you start with one here, and then you move down the chain, okay? And in the most common nomenclature, what you would do is that you would call a fatty acid like this, that has 18 carbons, an 18 carbon fatty acid. And then the 0 here actually means it has no double bonds. So an 18-0 fatty acid has 18 carbons and 0 double bonds. So if I would show you a structure like this, you should be easily be able to tell me what the nomenclature is, 18-0. Okay. Now for the unsaturated fatty acids, so those that are containing double bonds, we're again distinguishing two different kinds. We're distinguishing the monounsaturated fatty acids, MUFAs, that have one double bond. So now if you look right at the structure here, here's your terminal carboxy group, you're moving down the hydrocarbon chain, and right here in the middle, you have a double bond. Okay. So this, what we would call, again, we have 18 carbons, we have one double bond, so that's an 18-1 fatty acid. And if you want to be more precise and actually determine exactly where the double bond is, if you go down here, here's your carbon 1, Here your carbon's 2 to 8, and then here at carbon 9, you have the double bond sitting here. So you would call this an 18-1 delta 9. Again, starting to count from the carboxy group. There are multiple of these with different chain lengths. That's the same is true for the polyunsaturated fatty acids, and so these now have actually 2 or more double bonds in them. Okay. Here's your example, right? We're starting at the carboxy group, moving down. You have a first double bond here at C9. Moving further down, you have a second double bond here at the carbon 12. So what we would call this is an 18 carbon fatty acid with 2 double bonds. That's an 18-2. And they are at positions 9 and positions 12, so 18-2, delta 9-12. One important aspect that I would ask you to remember here is that these double bonds are almost always not conjugated in a fatty acid. Not conjugated, and I highlight the difference down here between conjugated and non-conjugated, means that the double bonds are actually separated by one methylene group. Okay? It turns out that actually in most fatty acids, you will find that the double bonds are positioned at C9, C12, and C15. Okay. So these are the ones that we will be seeing. They're obviously exceptions, but we won't really be encountering them this quarter. Okay. So for the purpose of our course, this quarter, since we are online, I will not obviously ask you to draw any of these. What I would ask you is that, for example, if you're given a structure like this, that you would be able to tell me the nomenclature for this fatty acid. There actually is also an alternate nomenclature for this, and you might be more familiar with this one. It's called the omega system. So you'll see this if you might go to traders and you buy your dietary supplements, like this one here, right? Omega-3 fish oil. This also actually is a fatty acid nomenclature that can be used. The difference here is that you start at the end. So at the methyl group opposite to our carboxy group, right? Omega, the last letter in the Greek alphabet, that's how you can remember it. You start at the end. So you start counting your carbons this way, and then you look at where your double bonds are positioned, right? Starting here, 1, 2, 3, first double bond, 4, 5, 6, second double bond. So using this nomenclature system, you would call this an omega-3, 6 fatty acid. Right? This is actually what this means on your supplement. It simply defines what kind of fatty acid you have in the supplement. A little bit deeper into what these double bonds actually mean physiologically, right? It's not just a chemical oddity, they actually have functional meaning. And so if you look on the left here, what I highlight here are saturated fatty acids, so no double bonds. They actually typically occur as pretty linear kind of molecules here, right? But the moment you actually introduce these double bonds, you're getting sort of a bending of the molecule. We call it a kink, right? And if you actually follow some of these illustrations here of the fatty acids in their space fill illustration, right? Here you have one double bond, so you have a little bit of a kink. Here you have 2 double bonds, a bit more, 3, even more. And here's one with actually 4 double bonds. So these start to kink more and more and more. Right? And so this 3-dimensional structure, whether they are more linear or really starting to have this kink, has a really fundamental impact on how we can utilize fatty acids and lipids and how we can store them. Okay. Another importance actually is the stereochemistry of these bonds, right? It's not just the presence and the number, but also the stereochemistry. What we can compare here are cis and trans fatty acids. And cis and trans here is relative to the stereochemistry of the double bond. And so I highlight this just here. Here's a cis double bond, and this is defined as having the hydrogen molecule atom sitting here in adjacent sides of the double bond, so both on the same side here. If you have a trans double bond, you have the hydrogen atoms on opposing sides of the double bond. Why is this important? This is actually important, again, for their chemical behavior. Okay. It turns actually out that if you have a trans stereochemistry of the double bond, you get a chemical property and a 3-dimensional structure of the molecule that is much more similar to saturated fatty acids. And only actually if you have a cis double bond, you get this kink. So stereochemistry really is crucially important here in this context. Why is this so important to us, this kinking in the molecule? If you actually think about trans fats in your diet, right, you're surely aware of it. There's lots of discussion about the health impacts of trans fats, is that you can imagine that whether you have a linear molecule or one that actually has one of these kinks, that it impacts how you can store them away, right? You can imagine, right, if you have sheets of paper, if they're nice and flat, you can stack them very neatly and put them away. But if you all like crumble them up, they start to be really big, right? And it's much more difficult to put them into the same space in your drawer. The same idea applies here. It is much harder to pack fatty acids tightly when they have this kink, okay? It also then has to do with accessibility, and this is likely what is behind the issue of trans fats, right? If you can pack them so tightly, it is likely becoming more difficult for an enzyme to actually break down these lipids. And so if you have too many trans fats, you have issues utilizing them, so they start to accumulate in your system, start to clog your system. There's a lot of ongoing research on trying to understand the physiological impacts and mechanisms behind this, but you should keep in mind, right, trans fats really have this part of issue. And because of that, right, you see on many of your nutritional fact sheets that people have to go very low on the content of trans fats, and sort of any food that you would buy that is deemed 0 or no trans fats has to be below this 0.5 gram trans fat per serving. That's the actual rule, right? It's not void of trans fats, but it has to be below this barrier. Okay. The bad news is trans fats are really rich in all of the yummy foods. All your fried food, all your sweets are typically quite rich in trans fats. All right, we talked a lot about fatty acids now and have defined their structure and properties, with some exception of where we use fatty acids as signaling molecules, for example, or as part of the breakdown pathways. Fatty acids are usually not existing in free form. They're usually esterified. Right? They are acids, so you don't really want them to accumulate necessarily in their free acid form. You want them to be conjugated with something that's esterified to something, and there are two major groups that we're looking at here. The first one are your triacylglycerides or TAGs. These are your neutral fats, right? They have many different functions. The major ones here is, we're already talking about it, right? They are good for energy storage. They're very inert. Neutral fats are very inert. They're are not very water soluble, so they're very good for packing and storing these molecules neat and tight. And so you can think about this, right? If you think about animals that hibernate, right? If you sort of go out into the national parks in the winter, all the bears are gone, they are hibernating. Before that, they're really fattening up. And the purpose of this is that fat can be stored very neatly and can get these animals through the winter without needing to take on any more dietary resources, right? And that's because fats can be packed so tightly and are such an energy-rich resource. The second function actually is that fats also have an insulating function from both heat and cold. And so this is what these guys are using, right? They have a huge layer of blubber, and this is what actually is keeping them warm, even though they are living in ice cold waters. That actually doesn't affect them as much because they have this huge fat layer as a matter of insulation. And the last one here is also a protective cushion. So you actually have a lot of fat between your organs that is meant to protect your organs from damage as a protective layer. So this is true for your neutral fats. Then we actually have another group that is important, and these are our phospholipids. They have a slightly different chemical structure and chemical properties, and these are the ones that are really critical for the formation of membranes. So these are the ones we want to be looking at today. Let's start with how they actually esterify, right? again, we have our tags and our phospholipids. The difference actually to some degree is in the composition of the type of fatty acids that are used, but the major difference really is how they are being esterified and the molecules we used as a backbone for this esterification. In your storage lipids, so the neutral fats, they're always esterified to a molecule of glycerol. So you see it right here. Here's your molecule of glycerol. I show the structure right here, right? We have a hydrocarbon backbone, 3 hydroxy functions. So they're ideal to now esterify 3 fatty acids to this backbone. So each of these hydroxy groups can be esterified with one of the terminal carboxy group of a fatty acid. So removing the carboxy functions through this ester bond, and we can make a neutral fat made up out of 1 molecule of glycerol and 3 fatty acids. Our phospholipids are a little bit more diverse. They can actually have different backbones and different conjugations. I just show 2 sort of major examples here. One would be our glycerol phospholipids. Here again, we're using a glycerol backbone, but we only esterify 2 fatty acids here. And then we have another head group here, which is bound through a phosphoryl group to the glycerol and is using an alcohol function. So this is obviously more polar, right? We have an alcohol here now. We'll get back to that in just a minute. But we also can have other backbones such as this here, which is thingosine. I show the structure here. You do not need to memorize it. I will not ask you about the structure, just to illustrate its difference to glycerol here. In this case, actually, we only have 1 fatty acid esterified. And again, through phosphor esters, we have a molecule of choline here, and we'll see this just in a second. So there's a different arrangement here in how they're esterified using the backbone and the number of fatty acids as compared to our neutral fats. Here's to highlight the structure now of our neutral fats. Again, up here is your backbone of glycerol, right? And here I have now 3 fatty acids, right, that are esterified with their terminal carboxy group to the hydroxy. So this is the remainder of the hydroxy group. Here's the carboxy group coming in. You're having water leave, and you can esterify the fatty acid to the backbone of glycerol. You do the same for the other fatty acids here. You get your tag. Okay. We can also highlight here, right? We have 2 saturated fatty acids here, 1 unsaturated fatty acids, and you can already see here in this space fill model, right, how much having these kinks that are generated by the double bonds in the molecule impact the structure of your tag. And you can imagine now, right, that this will push the molecule to be much wider, and an enzyme would have it easier to now attack this fatty acid for breakdown as compared to just having basically a neatly packed tag of 3 saturated, more linear type of fatty acids. How do these compare now to the phospholipids? Oh, before I go there, there's one more point here to this packing of the tags. So I highlighted here, right here would be an example now of our linear, our saturated fatty acids esterified to glycerol. And here's one where we have fatty acids with a number of double bonds that start this kinking. And the impact is really clear if you actually look at, for example, where you would find these tags. These ones here, more saturated fats and tags, actually are fat that we are producing, animals are producing. And so under room temperature, they would be solid. So you can think, for example, of trans fats from animals, butter, for example. And these ones here that are more unsaturated, they actually at room temperature will be in oil. And these are typically the tags you find in plants, right? And now you can think about why this might be important. Why would plants produce tags that have more of a liquid consistence under room temperature? And think about how we generate heat, right? These fats in animal systems, right, we can maintain our heat through our own system. Plants cannot, right? They're dependent on the outside temperature. And so having tags now that are in this is actually crucially important for their utilization, but also using these unsaturated fatty acids for the production of phospholipids impacts the integrity of their membranes. These membranes are actually much more fluid and are much more adjustable to different temperatures. Okay. So you can see the apparent advantage of using different kinds of fatty acids and how important these double bonds are for the consistency of the phospholipids, especially to adapt to different living conditions. So now to the phospholipids and looking at their structure compared to our neutral fats, right? Again, we have our fatty acids here, esterified to the backbone, right? In this case, we actually used glycerol here, but what we've done is we esterified only 2 fatty acids, and instead we have this polar head group here, right? And in this case, this is choline. Again, I'd not ask you to memorize its structure. What I want to highlight is, right, that this is attached to the glycerol backbone with a phosphate group, and the choline backbone here actually has a partial positive charge. So it makes it for a polar function. It's not neutral anymore, it's polar. We call it the polar head. And this combination of the neutral fatty acid tails and the polar head is really what chemically drives the formation of membranes. Okay. Because of the hydrophobic nature of the polar head, we will actually always try to face away from each other as much as they can. And because of the hydrophobic nature of the side tails here, they are actually attracted to each other. So we can generate these bipolar layers, right? So these fatty acids can form these bilayers of membranes because of their nature of having one polar side chain and one hydrophobic side chain. Fat in us, what does this mean? How is it important to us? Right? We had already talked about some of the importance of neutral fats. And so also within your system, we actually have 2 types of neutral fats and 2 functions, right? So your tags and your neutral fat, one actually is subcutaneous, so that's under your skin, right? And the other one is a visceral fat, and that's what you find, for example, in your adipose or between organs. So it's highlighted here, right? In red, this is your muscle tissue, here all your organs, and you can see there's a lot of neutral fats sort of highlighted in this tan color here that is covering your organs and is protecting them from damage. Now, if we zoom in to what it actually looks like at the cellular level, so now we're sort of going into the microscope, we're looking actually at cells that contain these fats, okay? These are called lipocytes or adipocytes, depending on in what tissue they are. And what you actually can see is here, right, these droplets here, actually in the cytosol, they're forming large oil bodies that are all together, and they're actually pushing all the rest of the cell contents here to the border of the cell. So they're expanding more and more, having this fat reservoir in here. And so in a microscope image, you can actually see that almost all of the cell is actually filled with this lipid droplet here, and only here at the very side, you have the remainder of the cytoplasm and the nucleus. So they're almost completely filled with fats and with oil. In plants, this actually is occurring heavily in the seeds. So seeds are a major storage. We had already discussed about them containing starch, but the most part of energy storage in seeds is actually lipids, okay? And they use this for germination. So again, for the growth of the plant, before it actually sees the light, before it can do photosynthesis. And this is the reason why we can make so many yummy oils like olive oil, canola oil, grapeseed oil, everything you can buy that you use for cooking. The reason is that seeds are such a good storage for oil, which plants use for their own growth and germination purposes. So this completes our dissection of the chemical properties and the functional importance of fatty acids and lipids. Watch out for the other part of the video for this lecture, where we're looking at how we're actually going to break down lipids and fatty acids for generating chemical energy.